Well, good afternoon, and thanks for staying, everybody. So it's my pleasure to present to you in about 10 minutes what we're doing in Seattle, Washington to realize this vision of what Lee calls P4 medicine. So Integrated Diagnostics was really formed 2 years ago to redefine in vitro diagnostics. So we didn't want to do something, a better way to measure known things, which is important. We wanted to find new things to measure to address the most unmet critical needs that we could define in medicine. And so we've taken a systems biology approach for earlier disease detection. And of course, systems biology is something that Lee Hood founded and is now becoming part of the curriculum in most graduate schools in the biology sector. We're doing highly multiplexed blood-based assays, and we have two major clinical targets. I'll speak to you a little bit about the lung cancer project. So we have lung in clinical development and also Alzheimer's disease early detection in development. We have a very high-tech platform. It is so high-tech we're going to use a CLIA model, as Bill alluded to, because we have very expensive instrumentation that requires great expertise to run. But in diagnostic medicine, it's not so much about the instruments. Companies provide answers for physicians, and that's what we're in the business of. So we're about 2 years old. We were financed, led by Interwest, $30 million financing, a white paper financing, which is very unusual in this day and age. Today in clinical development, and we will be commercial next year at this time. So let me give you a quote from Lee Hood, who is a visionary and also a very practical physician scientist. So, current diagnostics really look at a few biomarkers, and that's the case today, whereas ours can measure and have the ability to measure hundreds simultaneously. And you may wonder why that's really important. Well, the reason it's important is because it allows us to interrogate really the biological networks that are manifest in the proteome, which is the total expressed proteins in the blood. And so this is a very powerful statement. In the old days, we'd measure 1 or 2 proteins to try and make a diagnosis or an adjunct diagnosis. But we're able, using our mass spectroscopy technology, to actually do an unbiased survey and range from normal all the way to disease and look at what are the perturbations that are present in those patients. And we do this on one platform, mass spectroscopy, and we use a variant of it called multiple reaction monitoring. And this allows us to measure hundreds of proteins quantitatively simultaneously. And we also are avoiding one of the great problems going from discovery through commercialization, which is changing your analytic platform. And I've been caught in that problem several times, as others in the room may have too. But we're able to do our discovery and then drive all the way through commercialization on the same platform. So we have robustness and stability built into this. So when you have a platform, people don't invest in platforms. They invest in the future value of the products that emerge from the platform. And we spent a lot of time in the early days, before we got deep into the clinic, in identifying the indications that we wanted to do, and also doing the pharmacoeconomic modeling at the get-go so that we could understand how much we could spend and whether or not this was going to be viable and how long would it take us to get reimbursed for something like this. And so this is the first indication. And we don't have a name for it yet. It's a little kludgy right now, but it's a lung nodule molecular diagnostic test. And I was a little bit surprised when we really uncovered this problem, because there are over 3 million cases every year in the United States of nodules that get referred to a pulmonologist. And the pulmonologist has the history of the patient, and he has a CT image, just like that. And that's it. So the question is it lung cancer? Or is it not lung cancer? Is surgery needed? And there are a lot of costs associated with that, and a lot of very serious medical outcomes associated with that. And so this is really the algorithm that a pulmonologist uses. And you can see you have the CT up there, and then you see inserted the Integrated Diagnostics lung nodule test. So there'll be an in vitro diagnostic test that will give a result that says high probability of cancer, low probability of cancer. And that data, along with history and the CT image, will help sort where this patient goes in this continuum of risk, all the way from the lowest risk, which is watchful waiting, which basically is every 3 to 6 months follow-up CT and come back and we assess whether it's grown or not. So you can see that's a very risky move. Then there's the intermediate risk, which is, wow, maybe we'll do a PET scan. Okay, those are very nonspecific, but they're important. Or maybe we'll do a fine needle aspirate. Or maybe we can get to this lesion so we can do a bronchoscopic exam. Or if we're really concerned, we're going to go to a VATS procedure, which is a $27,000 procedure. And of course, anytime you're in the chest, you know you've got a problem. So there's a lot of morbidity and mortality associated with taking these decision paths. So I can't go into the clinical trials today. We don't have enough time. But this is one of the most heavily researched clinical trial that we know of in this space. We have exhausted virtually all of the pedigreed specimens in North America to address this question. And what I can tell you is that we've discovered through our clinical work about 345 proteins that are differentially expressed. And if you look at the chart, what we have done is we've sequenced these proteins, we go back to look where they originated from in the genome, and then we found what circuitry they're involved with. So it's really a backwards look and an unbiased backwards look because you only want to know the circuits that are perturbated. And if you were to sum up all of these different proteins, they would come out to be greater than 345 because many of the proteins are shared in those pathways. But on your right side, what you see are the results of about 1 million calculations of all these different so-called transition points. These are the basic data that comes off a mass spec. And this is a plot on the left axis of sensitivity, the ability to detect disease, and on the bottom axis, 1 minus specificity. And that line, which is an algorithmic development of 16 of the most informative proteins that act the best together, as well as 6 proteins that allow us to stably go across patients, which is a great failure of proteomics in the past. And you'll see the area under the curve in this receiver operator curve is approaching 0.85. So perfection is 1, virtually no test is there, and an acceptable diagnostic adjunct test in this area falls in at about 0.75. So I'm showing you early data. It's based on about 242 pedigreed patients. We'll have another 300 patients complete. We're also in prospective trials now. These are all retrospectively derived and fully pathologically based. So we're very encouraged by these data. These are better than we thought we would get. And we do believe that as we continue the algorithm development, that we will approach an area under the curve of about 0.9. Now, that's a very broad look at diagnostics, and I must tell you that that's not something a physician would use to derive whether or not the test works. But this gives us the fundamentals to set the cutoff bars such that we can drive either negative or positive predictive values, which are the most important probabilistic elements that a physician would use here. Just wanted to briefly show you our team. This is a team that has well over 100 years of diagnostics experience. Fortunately, it's not just my experience. And we have co-founders Lee Hood, all notables in the biotech space. They've done this before. They're serial entrepreneurs. And our investors also. You've heard today that your investors make a huge difference. And this is my 3rd venture-backed company, and I can tell you that they make all the difference in the world. Interwest is a terrific lead. We're also blessed with 2 evergreen funds, the Wellcome Trust and Biotech Cube, which is a sovereign fund from Luxembourg. And finally, our advisors, we have people that have been there before. So Greg Critchfield is really the medical pioneer behind BRCA1 and 2 at Myriad Genetics. He's on our board. And Milenko Tanzevich is the head of clinical labs at Brigham and Women's and Dana-Farber. So in summary, 2 years into it, we believe we have breakthrough technology and a first indication that should prove to be extremely valuable for the medical community as well as for our investors. We will be launching this time next year, It'll be a CLIA reference lab model. So like you heard from Bill Quirk, we believe in that model. This is a physician-to-physician test model. That's why the FDA sometimes goes into the periphery, but they seldom come into it unless you're doing something really wrong. And our follow-on product, which is early detection of Alzheimer's, is running about a year behind this product in an early clinical development. So thank you very much.